This is a brief introduction for our second paper that we'll discuss in phages this semester, titled Engineered Bacteriophages for Treatment of a Patient with a Disseminated Drug-Resistant Mycobacterium abscessus. A couple quick technical notes before we start. So supplementary data is data that is presented outside the formal body of a paper. As scientific journals have moved to publish more of their work online, they have increasingly moved a lot of the actual data in a paper to these supplementary data, and that is available exclusively via online, and that lets them save money on printing paper. However, it also makes a lot of the data available only in the separate part of the paper that you also should read if you want to understand all the data. Right? So the supplementary data, even though it's not in the formal body of the paper, it's still important for understanding what the authors are presenting. Another quick note too is that there's no formal rule that figures need to be presented in the order they're discussed in the paper. It's really nice if they are, but in this paper they do discuss figure 1 first, but then move on to figure 2 and then go back to figure 1 again. And so you should take a note of that as you work your way through the paper itself. So this paper is a bit more complicated than our previous paper. So I'll do the introduction in 2 parts. In this first part, we'll talk about the patient in this paper. So Isabel Carnell Holdaway is a teenager who lives in Great Britain, and she is pictured here third from the left, along with 2 of the authors on this paper, Dr. Helen Spencer and Dr. Graham Hatfull, and her mother. So Isabel suffers from cystic fibrosis, which is a genetic disorder that affects a number of organs in the body, including the lungs, digestive systems, and other parts. So what are particularly affected by CF are cells that produce mucus and sweat and digestive juices. Especially patients who have CF produce excess and thick, sticky mucus inside of their respiratory tract. So normally, your respiratory tract produces mucus to trap bacteria, other microbes, dust, and debris, and then the mucus is pushed out of your airways by a number of ciliated cells that line the tract that we call the mucus excretory system. So the mucus excretory system, these ciliated cells, the cilia, which are these long fibers that line the cells, they push the mucus up out of your tract, and you either cough or spit that out, or sometimes swallow it into your digestive system. And so that removes harmful microbes and other debris that get into your lungs when you breathe. Patients who have CF, the thick, sticky mucus can't be effectively removed by the mucus excretory system. And because it's stuck in the respiratory tract for a long period of time, that gives things like pathogenic bacteria a chance to build up in those airways. So patients who have CF frequently have respiratory infections, and this is a main complication of this disorder. So cystic fibrosis is a genetic disease, and so all 50 states and the District of Columbia in the United States screen newborns for this genetic condition. There are a number of signs that parents can recognize CF in their children, including their salt, their sweat is especially salty. They also might have a persistent cough, and you might see the thick mucus when they expire. So the molecular basis of cystic fibrosis disease involves the cystic fibrosis transmembrane protein, which is abbreviated CFTR. So the CFTR proteins are located in the cell membrane. So this is a protein that is part of our human cell membranes. So everybody has the CFTR protein. Right? And it's important because these proteins form a channel in our cells that are necessary for regulating fluid and the ion chlorine levels inside and outside of the cell. So we'll discuss more about the cell membrane in Unit 4, but in this current unit that we're working on, we are thinking about the different chemical properties of biological molecules. And cell membranes are made of hydrophobic chemicals called phospholipids. So the cell membrane, because it is hydrophobic, because the tails of the phospholipids are very nonpolar, it creates a separation, a barrier between watery layers outside and inside the cell. So some molecules are able to move through this barrier if they are small and nonpolar, such as gases like carbon dioxide. However, large, polar, and charged molecules, because they are hydrophilic, because they do not interact well with the nonpolar hydrophobic phospholipid tails, they do not move easily across the membrane. And so protein channels in the membrane are necessary for selectively allowing things like ions in and out of the cell. So the cystic fibrosis transmembrane protein genes are encoded in everyone's DNA. So every person has 2 copies of each of these genes in their cells. You inherit one of those copies from your mother and one of those copies from your father. Now, having just one working copy of the CFTR gene allows enough regulation of ion flow inside and outside the cell for a person to be healthy. Right? So a person can have one healthy copy of the CFTR gene and one disease copy of the CFTR gene, and they will not have cystic fibrosis. We will call these people a heterozygous carrier. But a person who has 2 non-working copies of the genes, they will have cystic fibrosis because there is not proper regulation of chloride ion transport inside and out of the cell. So the cystic fibrosis protein is encoded in the cystic fibrosis transmembrane, the CFTR gene, which is a nucleotide sequence inside of our genomes. There are over 17,000 different mutations in the CFTR gene that can cause cystic fibrosis. So even though the protein is not working, the specific way it's not working is a little bit different in many different patients. However, there are some common CF mutations, and the most common mutation is a mutation called delta F508. In this variation of the nucleotide sequence, 3 nucleotides are deleted in the middle of the CFTR gene, the nucleotides C, T, and T, and that causes the amino acid sequence that is translated from this gene to lose one of the amino acids. This deletion of amino acid from the protein causes the protein to be misfolded. Instead of being inserted in the cell membrane where it can regulate the flow of ions in and out of the cell, that protein becomes misfolded. And when it's misfolded, it gets shuttled to a garbage disposal system inside of the cell called the proteasome. The proteasome will degrade that misfolded protein, but also those cells won't have any functioning CFTR transmembrane ion channel. So patients with CF have a number of pathologies, including reduced lung function. So we measure the function of lungs in CF patients by measuring the forced expiratory volume, or FEV. And so if you want to measure how well somebody can breathe, you can have them breathe into this device that measures the pressure of air that they are able to breathe in and breathe out. Here are a few questions to consider when you are reading about Isabel and her condition. How old was Isabel at the time of this particular study? Did she have this delta F508 mutation? And what were some of the medical issues that Isabel was facing at the time of this phage intervention? And how are those issues in particular related to her cystic fibrosis